This lecture is going to be the entire lecture over neurophysiology. In this overview, what we're going to learn about is the parts of the neuron and the activity of each part. So the regional differences we call the receptive region, the conducting region, and the transmissive segment. Segment, region, doesn't really matter. The receptive region is the part where stimuli can be received. And for most neurons, that's mostly going to be the dendrites, but also the cell body of certain neurons. The conducting region is the axon. The initial segment is called the axon hillock. We call it the trigger zone because that's where an action potential is going to start. A nerve impulse runs down the axon. When it gets to the transmissive segment, hey, that's just the synaptic knobs or synaptic terminals where neurotransmitter is going to be secreted. The parts of those different regions of the neuron that matter are the membranes, and those membranes create electrical potentials, and any part of the neuron can have a resting membrane potential, but we're going to see regional differences other than that. So the local potential occurs at the receptive region, and the action potential runs down the axon. So the receptive region, hey, that's the part that receives the stimulus. It's the part that you can either hit with neurotransmitter, or if it's a sensory neuron, you can stretch it or whatever. However you stimulate it, that's where it's going to be stimulated. In general, that's the dendrites, and the potentials that are generated there we call local potentials because they don't keep going. They die out. The conducting region is going to be the axons. However long the axons are, any branches of the axons, all part of the axons, can conduct a nerve impulse. And then the transmissive segment is the part that releases neurotransmitter. So here's a diagram of a multipolar neuron showing you that not just the dendrites but the cell body is part of the receptive region. The initial segment is the trigger zone or axon hillock. In this case, the conductive segment is going to carry the impulse. We're going to combine initial segment and conductive segment into basically one thing, and then the synaptic knobs for the transmissive segment. So here you see a diagram showing you the different kinds of membrane channels and pumps that are found. So look on the left side first. The entire neuron has sodium-potassium pumps, which you already know. Pump 3 sodiums out for every 2 potassiums in, and then a scattering of sodium and potassium leak channels. We're pretty much going to ignore the leak channels. You just need to kind of in the back of your mind realize that they're there all the time. The gated channels are the ones that we care about because they're going to actually be stimulated to open or close. So in the receptive region, what opens and closes the gates, what opens them is the secretion of chemicals. And on a multipolar neuron, that's always got to be at a synapse. So it's going to be some kind of cation, just in general, and typically it's going to be a sodium ion or calcium ion or potassium ion, and then some of them are actually chloride channels. On the axon, whether it's at the axon hillock or in the conductive segment, you have two kinds of voltage gates. You have voltage gates for sodium and potassium, and voltage gates are opened by electrical current. So once you create the electrical current in the receptive region, that electricity opens these voltage gates. Here down at the synaptic terminal, you see that we are going to switch our signaler to calcium. We know that when you let calcium into glandular cells, that exocytosis, secretion can occur. So you see you have a voltage-gated calcium channel. And then to put the calcium back where you need it, you have a calcium pump. Hey, come on in. Okay, so we use the term potential for electrical current because it's a form of potential energy when it's stored in the form of concentration gradients for the ions. And of course, once it's flowing, then it's kinetic energy, it's electrical currency. So I don't know why they use the term potential. That's just the term they use. So again, you have a resting potential, a local potential, or an action potential. So here's a diagram of a part of the axon, and what you're seeing here is an axon at rest, and what I want you to look at first is the voltmeter. The voltmeter is registering -70 millivolts. That's a typical value for a resting neuron, any part of a resting neuron. And now let's look at what's going on in the membrane. So you see the sodium-potassium pumps are pushing 3 sodiums out. Those are the little purple circles, and pumping 2 potassiums in, and that's the little blue circles. Notice there's more potassium on the inside than there is on the outside, and there's more sodium on the outside than there is on the inside. And outside with the sodium you see chloride ions, and inside with the potassium you see PI, that's inorganic phosphate, and protein, and those are both trapped inside the cell, and they're anions. They have a negative charge. And of course you also have ATP. So our leak channels are just in equilibrium. With our pump, sodium sure can leak in and potassium can leak out, but the amount of leakage is very, very tiny compared to the ability of the pumps to push it back where they need it. Last thing you need to notice is that there's an electrical charge on the membrane. It's positive on the outside compared to the inside. Even though there are positive potassiums on the inside, there really aren't as many potassiums in as there are sodiums out. You've got those negative ions on the inside, and yes, you have negative chloride on the outside, but at balance, at rest, it's more negative on the inside. And this is just sort of a slight difference in electrical current just right across the membrane. Okay, so we did this in class. Draw a circle. See if you can label everything where it ought to be. So what your circle should look like, you should have sodium on the outside, potassium on the inside, chloride with the sodium, and protein with the potassium. I forgot to put the positive and negative signs, but we'll come back to that. So we know the sodium-potassium pump uses ATP to move 3 sodiums out and 2 potassiums in. You've got your leak channels, which we're kind of ignoring. And then there are actually 4 ions that have gated channels: sodium, potassium, chloride, and calcium, which at this point your book's been ignoring because it's not going to affect anything on the axon. All right, so here's a diagram that just shows you all the different kinds of membrane transporters. We'll look at them one by one. So here's your sodium-potassium pump, and you absolutely have to remember 3 sodiums go out for every 2 potassiums in. This helps contribute to the imbalance because there are 3 out for every 2 in, and that helps make the inside more negative compared to the outside. The leak channels are going to move things along their concentration gradients. Gated channels are going to open and close. That's the whole function of a gate. And here I expand on what the book has because I'm pulling in stuff on the sensory chapter. So your sensory receptors in your skin or your muscles or whatever might be mechanically regulated, and we call these mechanoreceptors. So if something gets touched or tapped or shaken or pulled or pushed, then the little gates open just because of the physical pressure. We'll come back and talk about those when we get to chapter 16. The chemically regulated gates, some chemical binds to its receptor, typically directly on the gate, and that includes not only neurotransmitters but also chemical senses, so your sense of taste, your sense of smell, and there's some chemical senses in the visceral body parts as well. The voltage regulated gates, these are the ones that are found only on axons. They're only associated with the action potential. So here you can see chemically regulated gated channels or chemical gates, and you can see that it opens when the neurotransmitter or the chemical binds. This particular one is a chemical gate for potassium, so the formal name is a chemically regulated gated potassium channel. And gates, like leak channels, still only allow diffusion, so the substance can only move along its concentration gradient. And here you see the voltage gate for sodium. Sodium's a little bit different because it actually has 2 different kinds of gates, an inactivation gate and an activation gate. The only time that sodium can flow through this gate is if both gates are open. So when you stimulate the sodium voltage gate, all the gates open and sodium flows. When it closes, initially it's the inactivation gate that closes, and then after that the activation gate will close and the inactivation gate will reopen. Okay, so we'll come back and talk about these because the sodium voltage gates are going to determine some other factors associated with membrane potentials. All right, now we're going to look at the ion distribution. ICF is intracellular fluid and ECF is extracellular fluid. So potassium is mostly concentrated on the inside, and it can cross through either leak channels or gated channels. But the anions in the ICF, those proteins and phosphate ions, are trapped. They can't leave the cell. And so there's always that negative core inside the cell. And then in the extracellular fluid, sodium can move across the membrane, chloride can move across the membrane, and the movement of those ions is going to determine your local inaction potentials. So at rest, remember we were sitting at -70, and you can see that in the voltmeter, the oscilloscope at the lower right side of this picture. And the inner side, the inner face of the membrane is negative and the outer face is positive. So we say the membrane is polarized. It has poles, a positive and negative pole. And so here you can see in the diagrams, let's see in the upper one, the resting membrane potential. Remember at rest the gates are closed. The membrane sits at -70 millivolts with most of the sodium and chloride on the outside and potassium mostly on the inside. When we depolarize, that is, we make the membrane less negative on the inside. Notice in our voltmeter it goes to from -70 to -60 millivolts. We open a gated channel for sodium, and sodium, of course, since it's mostly in the ECF, is going to flow in, and when it does, it brings the positive charges with it and makes the inside more positive, or we should say just less negative. So less negative is depolarization. And then if you open potassium channels or chloride channels, then those flow in a direction that makes the outside even more positive or the inside more negative. That hyperpolarizes the membrane, makes it from -70 to -80. All right, so if we graph that, depolarization means you're moving closer to zero from the inflow of sodium ions. Hyperpolarization is moving further away from zero, and that's from opening potassium ions or opening chloride ion channels. And in the case specifically of at synapses, this is what's going to happen. At the synapses, the neurotransmitter is released, it binds to the channels, it opens the gates of those channels. So if it opens sodium gates or even calcium gates, then positive flows in, it makes the inside less negative, and you get a little blip you see on the left. And we call that an EPSP, an excitatory postsynaptic potential, because it's a change at the synapse. And on the right, you're seeing hyperpolarization, an inhibitory postsynaptic potential, and that's from potassium or chloride. So in summary, we have these four ions, sodium, potassium, chloride, and calcium. And here you can see the direction that they diffuse. Notice that potassium is the only one that's going to diffuse out. But the effect on the membrane, chloride moving in has the same effect as potassium moving out. So both potassium and chloride inhibit, whereas sodium and calcium, both being positive ions on the outside, flow in, depolarize, and excite the cell. So that's the basis of our graded or local potential. These are the potentials that are found in the receptive region of the cells, the dendrites of sensory cells, or the dendrites and cell body of the multipolar cells at the synapses. So receptors give you receptor potentials. We're actually going to come back and talk about them separately in Chapter 16. And in this chapter, we're going to focus on postsynaptic potentials. So the receptor, remember, can be stimulated mechanically or chemically, and receptors you only excite. Now there's an exception to that, we're not worried about it, but in the retina of the eye, things are just very different. And then synapses can be either excitatory or inhibitory by opening potassium or chloride gates to inhibit. We call these potentials graded potentials because they can have different grades, and we grade things by weak or strong. And they can also be excitatory or inhibitory, which action potentials cannot do. So sodium and calcium are going to depolarize, and potassium and chloride can hyperpolarize. And the whole point of that is to move toward or away from threshold. So an excitatory local potential brings you toward threshold. If it hits threshold, it starts a nerve impulse. And sufficient stimulation that's excitatory by summation, by adding the effects of different stimuli together, can bring you to threshold. Inhibitory ones drop the polarity even more and take things further away from threshold. So we looked at summation when we studied muscle physiology. In lab, you got to stimulate a volunteer, and as you increase the voltage, the contraction got stronger, and that's an example of summation. So here we're seeing an example of summation in the nervous system where we're getting stronger and stronger and stronger stimuli, but each one is of short duration. That is, each one is over before the next one starts. So notice in the upper diagram, number 1, 2, 3, and 4, that each one of those little depolarizing currents drops back down to the resting membrane potential before you re-stimulate it. In the lower diagram, we're specifically not doing that. Our second stimulus comes on top of the first one. That is before the first one completely recovers. So in our top one, we have summation over space, and in the lower one, we have summation over time. So summation over space we call spatial summation, and here you can see as an example you've got multiple synapses labeled P1 through P5, and they're all hitting different parts of the cell body or the dendrites of this multipolar neuron. Look at the graph above, and you see that P1 through P5, all of them hitting at slightly different times, still manages to increase the electrical level up to threshold, and it has to be threshold at the axon hillock in order to matter. So that's spatial summation, multiple synapses. Temporal summation is hitting the same synapse multiple times. So here we're just using one synaptic knob labeled P2, but we're sending one stimulus after another before it can recover from the previous one. So you've got 5 different stimuli from the same synapse, and you get the same result. You reach threshold at the axon hillock. Okay, so we've gotten to the axon hillock, we've gotten to threshold. Now we're going to see how action potentials occur. So action potentials begin at the axon hillock. When it reaches threshold, they propagate down the axon, down the telodendria, all the way to the synaptic terminals. And action potentials use only voltage-regulated gates. So on axons, in general, that's the only kind of gates you have. And remember, there were only 2 choices there, potassium gates and sodium gates. And the whole idea of threshold only has to apply at the axon hillock. So if it's subthreshold, you don't get a nerve impulse. If it's threshold, you get 1 nerve impulse. Suprathreshold is just going to stimulate a string of action potentials, a whole little group of nerve impulses. So here you see a diagram showing you what's happening along an axon. Remember, these are voltage gates. So in our sodium gate, if you can read the one at the top, you have an activation gate and an inactivation gate. And when you start the beginning of the action potential, the activation gate is closed, but the inactivation gate is open. And the potassium gates are all closed at this point. At step 2, we've opened the activation gates. That then allows sodium to flow. Notice in this diagram that potassium channels also can open, but they don't open a whole lot, and they don't counteract the level of sodium, so we pretty much ignore them. Sodium diffuses out of the cell. That completely depolarizes it. And when it reaches, look at the little graph, when it reaches its peak of that little spike, then the inactivation gates close. Look at step 3, and now the potassium channels swing wide open. So sodium cannot diffuse, but potassium can. Potassium flows out of the cell, repolarizing the cell and bringing the potential back down, not just to where it started at -70, but overshooting down to, say, -90. And this gives us a period we call the hyperpolarization. And at the end of this, when it's hyperpolarized, the potassium gates, they're kind of on a timer, and they stay open for a very short period of time, and then they close. And then we're back to our resting membrane potential again. So if you look at one action potential, the graph of one action potential, here you see from the resting membrane potential at the onset of stimulation, we have our local potentials. They're summing up, so we're seeing summation at step 2. At step 3, we've hit threshold, and sodium is diffusing madly into the cell, making it more positive. Notice it goes past 0. This is called reverse polarization. It goes up to about positive 30, and as it hits its peak, those sodium inactivation gates close. The potassium gates are wide open. Potassium flows out of the cell, repolarizing it at step 4. And then at step 5, we have the overshoot. So it's hyperpolarized. It's more negative. It's farther from threshold until it finally recovers as the voltage potassium gates finish closing. All right, so if we wanted to do this, let's see if we can just draw a real quick sketch. And I am not very talented or patient when it comes to drawing neurons. So here is an axon. And I'm going to have local potentials on the receptive region. But once I get to the axon hillock, then I'm going to have voltage gates. And it's going to be voltage gates from there all the way down to the synaptic terminals. All right, next concept is the refractory period. A neuron is said to be refractory if you stimulate it and it doesn't respond. Refractory means resistant to stimulation. And we have 2 parts to the refractory period. It is absolutely refractory until the sodium activation gates are reset. And even after that, though, that's the period where it's hyperpolarized and further from threshold. So it's still harder than normal to get a stimulus response during the relative refractory period. So looking at this in dark purple, you have the absolute refractory period, and in lighter purple, the relative refractory period. That's the period when it's hyperpolarized. All right, propagation or conduction of the nerve impulse occurs by reinitiating or regenerating an action potential along that axon. So remember, these are voltage gates. So when voltage gates open, they create electrical current, and that electrical current can open the next voltage gates, which can then open the next voltage gates and the next one. So it maintains itself. It propagates the entire length of the axon down any branches it might have. It only goes from axon hillock out to synaptic terminals. It never goes in the opposite direction. And it's pretty much continuous. That is, those electrical currents are flowing from one end of the axon to the other. Here's the diagram out of your book showing you the voltage gates. And again, you've got everybody is closed on the far left, so that's the resting membrane, positive on the outside, negative on the inside. And then on the right, we've opened the activation gates. We're letting sodium flow into the cell. And that takes the electrical charge from negative 70 millivolts up to positive 30 millivolts. And then when the activation gates close, then we'll be back to rest again. So after the sodium gates do their thing, then the potassium gates are going to open. Potassium flows out, and that repolarizes the cell, resets the polarity back to being negative on the inside and positive on the outside. All right, so it turns out that not all axons are the same. Some of them are unmyelinated. We should have already known that. These tend to be pretty small, very small diameter, and they're usually associated with visceral pathways, that is from your internal organs, or general sensory or pain fibers. So just the feeling of pressure when you sit down or pressure on the bottoms of your feet, things that you tend to not pay a lot of attention to. And certain kinds of pain, not all kinds of pain. So if it's a dull pain or an aching pain, it's going to be in this category. Type C are the slowest. They conduct impulses at a speed of 1 meter per second. And here you see the continuous conduction on an unmyelinated axon. Every part of the axon has to go through that depolarization, repolarization sequence. On myelinated axons, because the Schwann cells, or in the central nervous system, the oligodendrocytes, are segmentally covering the axon with myelin. That electrically insulates the axon, and only those bare spots called the nodes in between adjacent myelinated cells are going to be able to have ions flow. So all your ion channels are there at the nodes. And the distance between one node and the next is pretty much exactly what it needs to be for the voltage created at one node to get to threshold at the next node. And so what you get is what we call saltatory propagation. The impulses leap over the myelin sheath from node to node to node, and it's a whole lot faster. It also saves a lot of energy because you're only having to have ions flow at these very tiny patches of membrane, and so you only need sodium potassium pumps at those spots as well. We find two kinds of myelinated axons, type A and B. A are big and type B are smaller. Not as small as type C, but still pretty small. And the bigger you are, the faster you are, and the more, if you have myelin, you're faster. So type A fibers are the fastest, up to 140 meters per second, and type B up to 18 meters per second. Type A are associated with your somatic senses, so your sensory information from the senses you're most acutely conscious of, and then your control of skeletal muscles. All of that is type A. Type B is visceral and general sensory, and again, that has to do with supplying your internal organs and also some general sensation. We'll talk more about the visceral supply when we get to the autonomic nervous system. So we'll come back to type B and C with the autonomic pathway. So here's a diagram showing you the saltatory conduction at one node. It's going to jump over that node to the next one. Okay, so we've got the group A's. The nerve type A is the fastest, up to this slide says up to 150, and then group B and C, the smaller ones, are the sensory and motor visceral ones. Okay, the frequency of action potentials is going to determine the brain's ability to interpret the strength of stimulation. So bear with me a second. We said that local potentials were graded. You could have weak ones or strong ones. Mostly by having the synapses re-stimulated, you release more neurotransmitter, and that gives you a stronger local potential. Action potentials, on the other hand, are all or none. If you open the voltage gates at threshold at the axon hillock, you get an action potential that goes to plus 30 every time. There's no difference in strength of action potentials. So how does your brain tell if it was a strong stimulus or a weak stimulus? And the answer is by the frequency. A stronger stimulus creates a higher frequency of action potentials. So here we have a diagram showing that. Our first is a subthreshold stimulus. Local potential did not get to threshold, no stimulation, no action potential. At threshold, you get one action potential, and the higher you go, the more action potentials you get. But notice they're coming closer and closer together until you get to the maximal stimulus. So the higher the action potential frequency, the more neurotransmitter is released at that axon, at the end of that axon, and the more neurotransmitter released, the stronger the local potential is on the next stimulated cell. So even if you have a series of 3 neurons in a pathway, you're still going to carry that message of how strong the original stimulus was. Okay, so as we get into synaptic transmission, here we're looking at initially the fact that there are 2 types of synapses. We have gap junctions, which are electrical synapses, and then our chemical synapses. Gap junctions we've already studied. We know there are gap junctions in smooth muscle, there are gap junctions in cardiac muscle, there are gap junctions between ciliated cells, and there are even gap junctions between astrocytes. It's simply a way for cells to directly communicate with each other, but because it's a connection between cells, we call it a synapse. As far as I know, neuron-to-neuron synapses do not fall into this category. There might be some minor exceptions, but none that I'm aware of. So all the synapses we're going to talk about are going to be chemical synapses. And we have two categories here. Ionotropic opens ion channels, and metabotropic stimulates metabolic changes in the cell and generally opens ion channels as well. Your book doesn't talk about the metabotropic ones here. It saves them till it gets to the endocrine system, but we need to consider both kinds. Okay, so just the basic parts. The neuron that is releasing the neurotransmitter is the presynaptic neuron, is the one coming into the synapse. And the synaptic cleft is where the neurotransmitter has to diffuse, and then the postsynaptic neuron is the one that has the receptors for the neurotransmitter. So at the synapse, when neurotransmitter is secreted, the receptors bind the neurotransmitter, and what happens next depends on the receptors. That is, it depends on the receiving cell, the postsynaptic cell. Those receptors could be part of a direct sodium channel, but they might also be part of a potassium channel. So since we know sodium excites and potassium inhibits, the effect of the neurotransmitter on the receiving cell is based on what receptors it happens to have associated with that. All right. In the presynaptic cell, when the action potential reaches the synaptic terminals, that's going to open voltage-regulated calcium gates. Calcium coming into the synaptic terminals triggers exocytosis of the synaptic vesicles, just a few at a time, dumping a little bit of neurotransmitter into the synaptic cleft, and then the calcium pumps in the synaptic terminals are going to quickly pump that calcium back out. So that the effect of one action potential might last a millisecond. Neurotransmitter diffuses across the cleft and binds to its receptor sites, and then it's quickly either deactivated or removed. So the effects, if it's a direct effect, that receptor that's binding the neurotransmitter is directly connected to the chemically regulated gate. This is the example of this is acetylcholine at neuromuscular junctions, directly open sodium channels. But in neurons, it depends. It could be excitatory or inhibitory. So here's the diagram from your book, and it's showing you, I think, yeah, this is figure 12, but it's showing you a neuromuscular junction. And you can tell that because those are muscle cells being stimulated there. It doesn't matter. They all work the same. So you see calcium channels opening. Those are voltage-regulated calcium channels when the action potential arrives. Calcium triggers exocytosis of the neurotransmitter out to the synaptic cleft, and then calcium pumps will pretty quickly pump that calcium back into storage and stop that process. But once the neurotransmitter is secreted into the synaptic cleft, it's going to bind to and directly open some ion channels. If it's indirect, the metabotropic effect, then the receptor is going to stimulate, the neurotransmitter still has to stimulate a receptor, but that receptor, instead of being part of an ion channel, is going to be part of a series of proteins that activate something we call the second messenger inside the cell. Best example of that is cyclic AMP, which is produced from ATP, and that's already more than you need to know. So the effect of a metabotropic neurotransmitter is that it can initiate protein synthesis or other long-term changes inside the cell, and also, by the way, open gates. So here's a diagram out of an older book, and what I want you to find is the neurotransmitter receptor on the far left of our close-up, and then the little green circle of norepinephrine binding to the neurotransmitter receptor, causing it to release its hold on something called a G protein. G proteins are used a lot. I never test over G proteins, but they're pretty ubiquitously used in the body. So the G protein always activates something. In this case, it's activating an enzyme that turns ATP into cyclic AMP, and cyclic AMP is one of those second messengers. It's just something that can activate a lot of other enzymes. So you see metabolic changes, you see gene transcription, you see enzyme synthesis. And if you go back up to the membrane, you see, oh, by the way, it also releases something that binds to the inside of the gate and opens it. And in this particular example, because it's binding to a sodium gate, it's going to excite the cell. Okay, we also have to be able to get rid of neurotransmitters. So there are 3 ways to do this. We can rapidly deactivate it with enzymes that are available all the time. We did this with acetylcholine. We had the enzyme acetylcholinesterase in the synapse just sitting there waiting for acetylcholine so that its effect is very short-lived. Our second choice is to pump it back into the presynaptic cell that secreted it. This takes a little longer, but it also saves neurotransmitter. It recycles neurotransmitter. So pumping it back in sometimes works with an enzyme. For example, with acetylcholine, you break it into acetyl and choline, but you pump the choline back into the cell that secretes it. So reabsorb it. And if there's not an enzyme breaking it down, that's going to be a little bit slower. And then our third choice is that if nothing else happens, it just diffuses out of the synaptic cleft and it's absorbed by nearby astrocytes. Okay, so we have different kinds of synapses. We're going to introduce them here. We'll come back to them with the autonomic nervous system. Cholinergic synapses are ones that use acetylcholine as the neurotransmitter. And we've already seen cholinergic synapses in skeletal muscle neuromuscular junctions. It's also pretty commonly used throughout the central nervous system and in the peripheral nervous system, parasympathetic and sympathetic. You don't have to know details of that now. We'll come back and do that with the autonomic chapter. It is direct. It could be excitatory or inhibitory depending on the cell that is being stimulated. It is removed by acetylcholinesterase, but the choline is reuptaken. Second important kind of synapse is an adrenergic synapse. Adrenergic synapses use the neurotransmitter norepinephrine, which has another name. It's called noradrenaline. And it's a chemical cousin of the hormone adrenaline, which is also called epinephrine. Norepinephrine is used in the central nervous system, but also in the autonomic, specifically in the sympathetic synapses. And again, we'll worry about sympathetic and parasympathetic later. But norepinephrine is metabotropic. It's indirect. It can be excitatory or inhibitory, so it still, remember, opens some kind of gates, but it works by a second messenger system. So it has some kind of metabolic effects on the cell as well. And then it can be pumped and then recycled, or there's a group of enzymes called monoamine oxidase, which can break it down. And in this case, it depends on where you are. So depending on the specific kind of synapse, it might be MAO. All chemical synapses have a couple of properties that you need to be aware of. Synaptic delay just means the transmission through a pathway is slowed down. So if I have one neuron with, say, a type A fiber going a certain distance, it's going to get there a lot faster than three neurons with synapses in between, because every time you hit a synapse, it's going to slow down the passageway. We've already looked at the effect of that when we studied parallel pathways, neural circuits. A second aspect of synapses is synaptic fatigue. You can actually deplete the amount of stored neurotransmitter. Honestly, I don't know how common this is. You can do it experimentally, and certain drugs can do that as well. All right, so as we go through the neurotransmitters themselves, please rely on the posted practice quizzes to make sure you know what you need to know about each one of these. Acetylcholine we've already met, and it's one of the two major neurotransmitters of the peripheral nervous system. Depending on where it's being excreted or secreted, it can be excitatory or inhibitory. We already know it's excitatory at skeletal muscle. Second, norepinephrine, that's the other one we already met. It's the second of the major neurotransmitters of the peripheral nervous system because it's also used in the autonomic pathways. Dopamine is a neurotransmitter found in the CNS. There are some PNS effects of dopamine, but nothing for us to worry about in terms of routine neural stimulation. So most of the dopamine effects we're going to see are in the central nervous system. Dopamine affects certain pathways in the brain that are found like in the reward circuit. So dopamine is actually a mood elevator. And cocaine works by blocking the reuptake of dopamine, causing dopamine initially to be in excess so you feel great. But then remember that neurotransmitter can diffuse out of synapses, and then it's lost. So the loss of dopamine long term makes your mood absolutely worse than even just you were normally before. Serotonin is another mood elevator. It's also a sleep inducer. Serotonin is found in, not serotonin itself, but its receptors are found in 5 different varieties, and it's found in a lot of different pathways. So it's hard to make general statements about serotonin, but we do use it for certain mood disorders, attention deficit disorder. We use what we call selective serotonin reuptake inhibitors, SSRIs. And they work like cocaine does on dopamine. They prevent the serotonin from being pumped out of the synapse, so that prolongs the effect of serotonin. Glutamate is an excitatory neurotransmitter. It always works by opening calcium gates, and it's found in certain pathways in the central nervous system, and it creates problems after a stroke. And the reason it does is because neurons die when you have a stroke, and a dead neuron tends to release all its calcium. Well, that excess of calcium can actually kill the next cell in the sequence. So if that next cell also dies, then it releases all of its glutamate, and that kills the next cell. So you have these little pathways of stroke damage associated with glutamate pathways. GABA is gamma aminobutyric acid. It always opens chloride gates, so it's always inhibitory. Stimulated by Valium, it's used to halt epileptic seizures. I just read an article this morning that was suggesting that taking a GABA supplement might reduce anxiety, but it cannot cross the blood-brain barrier. So there are other food supplements or herbal supplements that people take to try to increase the amount of GABA in their central nervous system, but I have no idea how effective that really is. Glycine is inhibitory in the spinal cord, and what it does is it prevents the reflex exaggeration. So if you stretch muscles, then the reflex reaction of that is to make them contract, but that just stretches the antagonistic muscle, which can make that one contract, and so you wind up having just spastic movements if glycine doesn't tamp that down. And that's how strychnine works, actually. It blocks the effect of glycine, and so it causes convulsions, and it can lead to death if it affects the respiratory muscles. Substance P is for pain. So substance P is a neurotransmitter. We've studied it extensively in the PNS. It's actually secreted in the PNS, and it excites pain pathways. So when things hurt, it's a chemical reaction. It's the secretion of this neurotransmitter in the pain pathways that the brain then interprets as pain. And substance P can be blocked. It's blocked by endorphins, which are the natural opiates of the central nervous system. So endorphins are similar to morphine and other opiates, and they block substance P, and that blocks pain impulses. And if you block them all, it's not like it's a mood elevator, but it does make you feel like you're floating, and it kind of makes you feel elated. Some gases are neurotransmitters. This is nitric oxide and carbon monoxide. And we know that they are working in both the CNS and the PNS, even though we don't entirely understand them. We know that nitric oxide is an important vasodilator. This is the way nitroglycerin tablets work to open coronary circulation in people with chest pains associated with their hearts. It's also the way Viagra works. Okay, so as we study neurophysiology of synaptic transmission, we're going to look at some problems. You're actually going to answer questions on the test about the problems associated with synaptic transmission. So where can things go wrong? I can have problems with the axon not sending the signal, or I can have problems at the synaptic terminal, or problems with the receptors, and I can have problems that are either excitatory or inhibitory. So again, this is a diagram out of another textbook, so let's work our way through it. So at the very top, if we block the membrane channels at the axon hillock, then we don't get a nerve impulse at all. No nerve impulse. If that's a motor neuron, you're going to have paralysis. If it's your respiratory muscles, you're going to die. TTX is tetrodotoxin. So that's the toxin associated with pufferfish, and people eat pufferfish as sushi. So there's always kind of a risk of TTX, tetrodotoxin. Lipid-soluble anesthetics can depress membrane sensitivity. This is going to reduce the impulses of the conduction of nerve impulses. So this is the way alcohol makes people sleepy. Demyelinated axons, arsenic and lead. These are very slow processes, but again, as we would see the same thing with demyelinating diseases like multiple sclerosis, without the myelin sheath, saltatory conduction won't occur, and the impulse just dies out. Once you get to the synapse, you can either block the release of neurotransmitter or increase it. You can block the receptors, or you can inactivate the neurotransmitter breakdown. So In this diagram, oh, and I skipped one at the top. The pink boxes are stimulatory and the blue boxes are inhibitory. So caffeine and theobromine, which is the relative of caffeine that's found in chocolate, depolarize the axon hillock. That is, they lower the threshold and makes it easier to send nerve impulses. All right, back to the synapse. If we block the release of neurotransmitter, no neurotransmitter secreted means you don't stimulate that synapse, and that's the way botulinum toxin works. It paralyzes muscles. Spider venom, I don't know which ones, but some spider venom can increase neurotransmitter release, and that's going to overstimulate the synapse. You can block the neurotransmitter from binding, like atropine does, or you can stimulate the receptors with a different chemical, like nicotine does, or you can block the inactivation of neurotransmitter, like anticholinesterase drugs do. Now, you're going to answer questions about these on the exam, but you don't have to memorize any of this information. Just go through it and make sure you understand what each one of these different possibilities are and whether they're going to excite or inhibit the synaptic transmission. Okay, so if I block the release of neurotransmitter, I inhibit the neurotransmitter. If I block the removal of neurotransmitter, I prolong the effect of neurotransmitter. If I block the receptors for the neurotransmitter, I inhibit the neurotransmitter effect. But if I bind to and mimic the neurotransmitter activity, then I prolong the neurotransmitter effect. So work through that logically, make sure it makes sense to you. All right, in our postsynaptic cells, we've already talked about this once, but we're coming back to it. You have excitatory postsynaptic potentials and inhibitory postsynaptic potentials, and you can have summation that is temporal or spatial or some combination thereof. We've kind of already been through this, so just work through summation. Make sure you understand spatial summation. You're stimulating multiple synapses at the same time. Temporal summation, you're re-stimulating the same synapse over and over again. So it's excitatory if it brings you closer to threshold, and it's inhibitory if it hyperpolarizes or pulls you farther from threshold. We can also stop things at the source by doing what we call presynaptic inhibition or facilitation. So when you have a synapse between one axon and another axon, it can either stimulate or inhibit the secretion of neurotransmitter at that. I have no good examples of that. I just know that it can be done. So if it's inhibitory, it slows or stops the secretion of neurotransmitter, and if it's excitatory, it's going to stimulate it. All right, some things that can affect how well neurons function. If you have an ionic disturbance in your extracellular fluids, then your membrane potentials may be messed up. So a high concentration of acid, that's a low pH, remember, inhibits, and a high concentration of base, a high pH, is going to stimulate excessive neuronal activity. So if you have acidosis and it's severe enough, you go into a coma. But if you have alkalosis, then you start having convulsions. Electrolyte concentrations can cause problems. Too much potassium can inhibit, and high calcium can excite. Even temperature changes can affect neuron excitability. When you have a fever, it can make your neurons more excitable and cause convulsions. And obviously there are drugs that can alter neuron function. So caffeine, we said, lowers the threshold, and anesthetics raise the threshold. Neurons have a very high metabolic rate, and so their mitochondria are very active all the time. They are totally dependent on not just oxygen but glucose delivery from the blood, and their high metabolism can shorten their life through oxidative stress, just the buildup of oxygen in the cells. But anything that cuts off the blood supply can kill neurons. Fatigue is something that's actually regulated through a system that involves adenosine as a signaling molecule. Remember, adenosine is the A in ATP. So what happens when neurons are very active is they start selectively letting some adenosine out of the cell. They don't do it because they don't need it anymore. They do it as a signal. The signal is to the nearby blood vessels to increase the availability of blood, but it also sends signals to the brain inducing fatigue, feelings of fatigue, or needing to sleep. The problem with adenosine is that its signal gets blocked by caffeine, and so this is why caffeine keeps you awake. Other problems associated with the blood supply to the brain. TIA is a temporary or transient ischemic attack. Ischemia is a lack of blood flow. So if anything just cuts down, doesn't have to stop it completely, but cuts down the flow of blood to the brain, that's ischemia, and that can cause death of some of the neurons, leading to a stroke. A stroke in general is caused by either a blood clot or a burst aneurysm, anything that completely and irrevocably stops the flow of blood. Okay, that's it. And if you have any questions, be sure to go through the chapter summary. Also, look at the practice quizzes and study your Learn Smart.